In a remote corner of what we now know as Siberia, about 90,000 years ago, the world was a very different place. Humanity was not a single species, but a mosaic of close relatives, some robust, others more graceful, all struggling to survive. In wild and changing landscapes in this setting, a young woman walks barefoot alongside a frozen river. The air, despite it being summer, bites the skin as if it were winter. She patiently searches for small shells among the stones. Perhaps she dreamed of turning them into ornaments, or perhaps it was simply a distraction. She doesn't find any. Upon returning to her home, the smoke from dinner rises from a cave. Freshly hunted deer meat, carefully collected roots and leaves. But her home is not ordinary, it is a cavern shared by beings of a peculiar appearance. Elongated skulls, wide and strong bodies. They are human, yes, but not like us. They are Neanderthals and Denisovans, and she is the daughter of both. A hybrid girl born from the crossing of two extinct branches of humanity. Her life was brief. She died before reaching adulthood. She left. No tools or art, only a bone fragment barely 2 centimeters long. However, that small piece found in the famous Denisova cave has revealed secrets that redefine our history. DNA analysis confirmed her mixed lineage and her age, at least 13 years old. Perhaps she fell ill. Perhaps she was a victim of hunger or a poorly healed wound. Today, we know her as Deni. And although her existence was reduced to a single bone, her story speaks for millions. It is living proof, though fossilized, of how intertwined and complex human history is. Although Deni's bone fragment may seem insignificant, its discovery marked the beginning of an even deeper story. And like any good story, it originates in a place that for centuries kept silent amidst shadows and ice. The Denisova cave, hidden in the valley of the same name in Siberia, is not just any cave. This stone shelter has witnessed thousands of years of human presence. In the 17th century, a hermit named Denis lived in its chambers. Ironically, without his knowledge, his name would end up designating not him, but an entire branch of humanity. In the 1970s, Russian paleontologist Nikolai Ovidov began the first modern excavations in search of cannid remains, but what would be unearthed later would exceed all expectations. To date, 22 archaeological layers have been identified, spanning from historical times to more than 200,000 years ago. It was not until 2008 that a group of Russian archaeologists found the bone of a child's finger among the sediments. At first, it was thought to be between 30,000 and 50,000 years old, but later studies adjusted the age to between 52,000 and 76,000 years. She was called X-Woman. Her mitochondrial DNA, inherited maternally, revealed something astonishing. She belonged neither to modern humans nor to Neanderthals, but to a completely different species. In 2019, new dating led by Greek archaeologist Katerina Duka revealed that the oldest individual at the site, Denisovan, lived between 123,000 and 195,000 years ago. And based on tools found at the site, it is believed that Denisovans already inhabited that cave more than 280,000 years ago. Human history is rarely as simple as we were told. Just as the Denisova cave revealed an unknown species, it also forces us to correct long-held misconceptions. For years, it was believed that Homo sapiens was the first human species to leave Africa and expand throughout the world. I myself once thought so, and you probably did too. But the reality is more complex. Other human species already inhabited vast regions of Europe and Asia long before our arrival. It is known, for example, that Neanderthals lived between 190,000 and 97,000 years ago, coinciding with the period when Denisovans also lived. Deni, the hybrid girl, lived between 90,000 and 100,000 years ago. If her mother was a Neanderthal, it is logical to think that they also occupied the cave at that time, perhaps from much earlier, but for now, those are the only confirmed dates. If Denisovans and Neanderthals shared their genetics, they also shared the same roof dock. To understand how they coexisted, we must look not only at their DNA, but also at the place they called home, the Denisova Cave. This rock shelter of approximately 270 square meters, about 2,900 square feet, offered much more than shade and stone. Situated 700 meters above sea level in a mountainous region that is still green and fertile, the cave overlooked a landscape of cold and imposing beauty. Just 28 meters away flowed a river, a vital source of fresh water and fish. It is not surprising that generation after generation decided to settle here. 100,000 years ago, the world was already a colder place. Although the last great glaciation, known as the Ice Age, had not yet begun, the climate required constant shelter. Cave like this could mean the difference between life and death. It is likely that hundreds of generations were born, laughed, hunted, and died within its walls. Some died of hunger, others were attacked by hyenas, and on lucky nights, they celebrated a successful hunt. Long before Homo sapiens crossed the African continent, Denisovans were already living in this shelter, and they were not alone. Neanderthals also occupied this space, possibly at the same time. Although they were different species, DNA shows they were similar enough to mate and have fertile offspring more than once. The physical environment of the Denisova cave provides a glimpse into the daily lives of its occupants, but it is the genetic data that has most clearly revealed who they were and how far they reached. The Denisovan genome contains approximately 17% Neanderthal DNA, the result of multiple interbreeding events between the two species. The genetic evidence demonstrates a close and sustained relationship over time. To date, 5 distinct Denisovan individuals have been identified in the Denisova cave: Denisova 2, 3, 4, 8, and 13. To these is added Denisova 11, known as Deni, the only confirmed hybrid between a Neanderthal and a Denisovan. The remains found include a finger bone, 3 molars, a partial jawbone, a limb fragment, and another from the skull. Most of the available information comes from ancient DNA analysis due to the fragmentary nature of the fossil record. For years, all known Denisovan fossils came from the same cave. In 2019, a second location was confirmed, the Baishia Karst Cave, on the Tibetan plateau, more than 2,700 kilometers away. A partial jawbone originally discovered by a Buddhist monk in 1980 was genetically identified as Denisovan. This demonstrates that the Denisovans were not a localized population, but a species with a wide distribution in Asia. Despite this expansion, the scarcity of complete fossils continues to limit knowledge about their morphology and way of life. Knowing where they lived and with whom they coexisted is only the beginning. To understand who the Denisovans really were, it is necessary to analyze their genetic information and their place in the human evolutionary tree. The mitochondrial DNA extracted from the individual Denisova 3 has helped to clarify part of the kinship between Denisovans, Neanderthals, and Homo sapiens. This type of DNA, which is inherited exclusively from the mother, allows geneticists to compare the number of different nitrogenous bases between species and estimate their genetic distance. The difference between modern humans and Neanderthals is approximately 202 bases out of a total of 16,500, a variation of 1.22%. Denisova 3, in contrast, shows a difference of 385 bases with respect to current human mitochondrial DNA, that is 2.33%. In comparison, chimpanzees show a difference of 1,462 bases, or 8.86%. These figures should not be interpreted as a scale of evolution or intelligence. A greater difference does not imply inferiority. In fact, Denisovans had unique and functional adaptations, a topic that will be developed later. The relationships between these 3 human groups are complex. Homo sapiens evolved mainly in Africa, while Neanderthals and Denisovans emerged in Eurasia. The genetic interactions between them along with their wide geographical distribution, mean that human evolution does not follow a straight line, but rather a web full of intersections. Branches. It is generally accepted that the human lineage separated from the common ancestor of Neanderthals and Denisovans about 600,000 years ago. Later, about 400,000 years ago, the two groups split. It has been proposed that Denisovans could be descended from an early migration of Homo erectus, or, more likely, from Homo heidelbergensis, which reached Asia. It is also possible that their origin comes from a mixture of both groups, because Denisovans, interbred with more archaic hominins like Homo erectus or other, as yet unidentified species. Their genome reflects a more ancient mixture. This genetic complexity means there is no single clear answer as to how exactly these species are related to each other. What seemed to be a single species may never have been one. As genetic studies advance, it becomes increasingly evident that the Denisovans were not a uniform group. Recent research has revealed that the Denisovans could have been composed of more than one genetically distinct population, possibly even multiple species. An international team with participation from the universities of Caveman, Indonesia, New Zealand, and Singapore collected DNA from 161 modern individuals on different islands in Indonesia and compared it with the genomes of current humans, Neanderthals, and Denisovans. Through inherited fragments of Denisovan DNA, scientists reconstructed genetic characteristics of these ancient populations. The results confirmed a much greater diversity than expected. In some Indonesian individuals, two types of Denisovan genomic signatures were detected, different both from each other and from the Denisovans found in Siberia. This explains many of the inconsistencies and complexities in their genetic profiles. Wide geographical distribution and multiple interbreeding events with other hominins explain this diversity. At least 3 large groups have been proposed, one in Siberia, Kazakhstan, and China, another in mainland Southeast Asia, Thailand, and Vietnam, and a third on the islands of Indonesia. These last 2 groups separated from the Siberian group more than 250,000 years ago, even before the appearance of anatomically modern humans. Of the lineages detected in Papua New Guinea, shows such a level of divergence that, according to geneticist Murray Cox of Massey University, New Zealand, it could be considered a distinct species, although there are no fossils to confirm it yet. Genetic data show that this group was as different from the Siberian Denisovans as it was from the Neanderthals. Everything indicates that there is still much to discover about these hominins. The possibility of fossils appearing in Southeast Asia remains open. The genetic history of the Denisovans not only includes interbreeding with Neanderthals and modern humans, but also involves a much older interbreeding event with an even more archaic human population. One of the most relevant discoveries in recent years is the identification of an interbreeding event that occurred more than 700,000 years ago between the common ancestors of Neanderthals and Denisovans and an even older human population known as super-archaic hominins. This event occurred before the separation between Neanderthals and Denisovans. The super-archaics were genetically more distant from the Neanderthal Denisovan ancestors than any other pair of human populations, with known evidence of interbreeding. For reference, modern humans and Neanderthals had at most about 750,000 years of separate evolution at the time of their interbreeding. In contrast, the super-archaics and the Neanderthal Denisovan ancestors had been separated for more than 1,000,000 years. Despite this extreme genetic distance, interbreeding occurred. It is estimated that up to 4% of the Denisovan genome comes from this still unidentified archaic species. This information expands the known limits of genetic compatibility between human populations, separated by vast temporal scales. It also raises a key question: how many other archaic human species, as yet unnamed or without identified fossils, could have contributed to the genome of the known species? By interbreeding with Neanderthals and Denisovans, modern humans inherited part of their genome. But beyond DNA, an inevitable question arises. What did the Denisovans look like physically? Although no complete skeletons have been found, the available fossils allow for the inference of some anatomical traits of the Denisovans. The finger bone found is almost indistinguishable from the same bone in Homo sapiens. In contrast, the molars are markedly different, large, robust, similar to those of archaic humans from the Middle Pleistocene. The third molar is larger than that of any Homo species, except habilis and rudolfensis, and is more reminiscent of Australopithecines. The second molar, larger than that of modern humans and Neanderthals, resembles that of Homo erectus and habilis, suggesting a diet based on fibrous vegetables, roots, fruits, nuts, meat, and marine resources. Large and powerful jaws were necessary to process this type of food. A fragment of a parietal bone shows similarities to Homo erectus. Using genetic methylation techniques, a facial reconstruction of a Denisovan has been achieved, indicating a wide and projecting face, a large nose, a sloping forehead, a prominent jaw, an elongated and flattened skull, and a broad chest and hips. Their body was compact, adapted to the cold, and probably shorter than that of modern humans. Their cranial capacity is not known with precision, but it is estimated to be similar to that of Neanderthals, who had larger brains than us, although this does not imply greater intelligence. The brain volume was linked to more developed senses, not a larger prefrontal cortex. They were not inferior beings. They were humans with the capacity for reasoning, logic, and curiosity. We share 99.98% of the same mitochondrial DNA. From their genes, we cannot know how they thought, but nothing indicates that they could not have asked the same existential questions as we do. They were different, not lesser. Although the Denisovan fossil record is limited, their genetic legacy is present in millions of people today. Their genes continue to speak for them, shaping physical traits, immune systems, and unique adaptations. It is accepted that Denisovans, Neanderthals, and modern humans had notable physical differences from each other. Some groups may have developed lighter skin to adapt to the scarcity of sunlight in cold climates. Others, perhaps, had epicanthic folds, like those observed today in East Asian populations. Although we do not have complete skeletons, reconstructions based on genetics suggest that Denisovans were similar to Neanderthals. Wide faces, large noses, prominent jaws, and robust bodies adapted to the cold. The Denisova cave has provided Middle Paleolithic tools such as scrapers, denticulate instruments, and lithic cores. Some deposits are over 287,000 years old. Upper Paleolithic objects were also found: a marble ring, an ivory ring, tooth pendants, and a chlorodolite bracelet. Although Denisovan occupation ended around 55,000 years ago, the authorship of these pieces has not yet been definitively determined. Denisovans interbred with modern humans, mainly in Oceania and Southeast Asia. Genetic studies have identified multiple introgression events. Populations such as Australian Aborigines, Papuans, Fijians, and Polynesians have high levels of Denisovan DNA, with percentages ranging from 1.9% to 6%. In contrast, East Asian and American populations show lower levels, around 0.2%. Some studies suggest at least 3 waves of introgression from different Denisovan groups, which reinforces the idea that they were not a homogeneous population. Several key adaptations in modern humans derive from Denisovan genes. The EPAS1 allele present in Tibetans improves the body's efficiency at high altitudes. In the Inuit, certain genes associated with body fat distribution also come from this introgression. In Papuans, inherited genes affect bone structures, immunity, and metabolism. A 2021 study detected a genetic segment in Latin Americans associated with lip thickness, inherited from archaic humans. This same trait appears in populations in Asia and America. These genetic differences do not imply superiority or inferiority. Having archaic ancestry does not define an individual's worth. All current humans, including sub-Saharan Africans, who have minimal levels, carry some amount of Neanderthal or Denisovan DNA. The Denisovans were highly adaptable. They survived in both the extreme cold of Siberia and the tropics of Southeast Asia. They did not know mathematics or writing, but they mastered their environment, created complex tools, and developed strategies to withstand the harshness of their world. Over hundreds of thousands of years, they hunted, migrated, resisted famines, and left a mark that we continue to discover in ourselves to this day. The extinction of the Altai Denisovans has no definitive explanation. We may never know for certain what caused their disappearance as a visible group in the fossil record, but genetically, their story did not end. Part of their legacy lives on in us. For a long time, it was thought they disappeared about 50,000 years ago, like many other human species. However, recent analyses indicate that some groups survived until as recently as 14,500 years ago. Genetic studies reveal that interbreeding with Homo sapiens continued until as recently as 15,000 years ago in some regions of Southeast Asia and Oceania. On mainland Asia, they may have been displaced, assimilated, or absorbed by modern human populations. But in remote areas of the Pacific, their genetic footprint persisted much longer. That is why some current populations, such as the Papuans or Australian Aborigines, retain significant proportions of Denisovan DNA. This raises a profound question. If they still live in our genes, can we say that the Denisovans are truly extinct? The idea that modern humans are separate from the rest of the natural world is a cultural, not a scientific, construct. Anthropocentrism has guided myths and narratives for centuries. But biology reminds us that we are part of a much broader and more complex evolutionary tree. This series seeks to reconnect that shared history, not to glorify the past, but to better understand who we are. It is amazing to think that such an important human species has left so few remains, while we have complete skeletons of extinct animals that are much older. But with just a bone fragment, we have rewritten entire chapters of our history. And that is just the beginning.